Today we'll explore how gut bacteria navigate the challenges posed by human-targeted drugs, or HTDs, which extend significantly beyond just traditional antibiotics. Based on the mini-review by De La Cuesta Zuluaga et al., we will examine the response, resistance, and recovery cycles that define microbial survival in the face of our medications. This sketchbook digest provides an accessible look at the complex, often overlooked interactions between common pharmaceuticals and the human microbiome. While we traditionally view antibiotics as the primary disruptors of our gut health, research by Meyer et al. Reveals that nearly a quarter of human-targeted drugs, such as statins and antipsychotics, also inhibit the growth of at least one gut bacterial species. This means that many common medications we use for non-infectious diseases may be unintentionally acting like antibiotics, potentially reshaping our microbiome in significant ways. Understanding these off-target effects is crucial as we move toward more personalized medicine and a deeper awareness of the second genome within us. When analyzing drug efficacy, we distinguish between a direct assault on a pathogen and indirect pathways that work through the host's own biology. Host-targeted drugs, or HTDs, are particularly effective because they utilize both of these mechanisms simultaneously to combat infection. By combining direct growth inhibition with altered host physiology, HTDs create a powerful two-pronged offensive against microbial threats. Today we'll explore the three primary mechanisms of direct assault non-antibiotic drugs exert on gut microbiota, starting with growth stop, which illustrates dose-dependent bacteriostatic effects. Moving to the leak, we see how common medications like NSAIDs cause physical membrane damage and oxidative stress, effectively compromising bacterial integrity. Finally, the imposter showcases molecular mimicry, where drugs like Prozac trick bacterial transporters by masquerading as nutrients to block essential uptake pathways. Think of the gut environment as a house being renovated by medications, which indirectly reshape the microbiome by fundamentally changing its physical living conditions. For instance, drugs can thin the protective mucus layer or raise pH levels, allowing oral microbes to migrate deeper into the digestive tract. Finally, medications like statins can alter bile acid profiles, creating inflammatory environments that ultimately dictate which bacterial species are able to survive. Think of a single cell as a fortress equipped with a versatile defense toolkit to survive drug encounters. We see mechanisms like the bouncer efflux pumps that kick out invaders, enzymatic scissors that shred drug molecules, and target modifications that essentially change the locks to prevent entry entirely. This dynamic toolkit explains how bacteria like B. thiothiomicron maintain their resilience against modern medicine. When direct resistance fails, bacteria employ hiding strategies like persistence and sporulation to survive antibiotic threats. Persistence is a reversible metabolic nap that can actually be triggered by antidepressants, while sporulation acts like a biological bunker, allowing species like C. difficile to survive for weeks. Recognizing these survival tactics is essential for understanding how infections can linger despite aggressive treatment. When we examine microbial community dynamics, we find that survival against toxic drugs often depends on emergent properties, where the group outcome differs significantly from that of individual species. Through mechanisms like cross-protection and cross-sensitization, microbes can either collectively shield each other from environmental threats or increase their shared vulnerability. Ultimately, this illustrates that the functional village of a microbiome creates a complex ecosystem where the whole is greater than the sum of its parts. Cross-protection, often called the human shield, occurs when a hero microbe reduces environmental drug concentrations through mechanisms like transformation or sequestration. By acting as a buffer, these heroes create a safe haven for sensitive neighbors that lack their own resistance. This isn't just a biological curiosity. Research shows it is a widespread phenomenon found in up to 26% of microbial community interactions. In this case study, we examine the sponge effect, where medications like entacapone bioaccumulate by chelating iron, effectively starving beneficial commensal bacteria. This disruption creates an environment where pathogens can thrive, highlighting a critical link between drug metabolism and gut health. Fortunately, we can reverse this imbalance through simple iron supplementation to restore the microbiome's natural diversity. In our discussion of cross-sensitization, it is vital to recognize toxic friends, safe drugs that produce harmful metabolites during processing. This factory metaphor illustrates how the metabolism of a benign substance can release smoke or toxic byproducts that damage nearby cells. While these events are rare, they are potentially dangerous and typically triggered by high drug concentrations. Horizontal gene transfer acts as a microbial "black market," with gut bacteria being 25 times more likely to swap genetic material than their environmental counterparts. For instance, Antidepressants like fluoxetine can stress these bacteria, prompting them to exchange efflux pump genes and develop resistance to antibiotics. This highlights a concerning, quote, collateral damage, where non-antibiotic drugs inadvertently drive the evolution of dangerous superbugs within our own bodies. When we look at the impact of drug exposure on microbiome health, the recovery process is far from a simple linear progression. It is an inherently ecological journey that depends on specific biological preconditions. As shown here, a higher baseline diversity acts as a safety net, allowing for a more resilient, quote, bounce back after a sharp decline in species richness. Crucially, the presence of dietary degraders, specifically fiber and mucin users, is essential to jumpstart the ecosystem and facilitate a return to equilibrium. This illustration highlights the social cure, demonstrating how social environments can fundamentally accelerate physical healing processes. While isolated subjects often show slow and erratic recovery, those co-housed with others heal much faster due to the transmission of beneficial microbes through physical contact. Ultimately, this research underscores the profound biological impact our social interactions have on our overall health and resilience. Our field notes reveal that host-targeted drugs can mimic antibiotics, unintentionally triggering cross-resistance within microbial communities through collaborative survival mechanisms. Since recovery depends on a diverse biological village, it is crucial that we reconsider how these treatments impact the overall ecosystem. Looking ahead, the goal for future therapy is to design drugs that respect the neighborhood by preserving the delicate balance of our internal microbiota.